To continue with our discussions, my dear students, so we have the structure of the bacteria known as the spores or endospores or bacterial spores. Usually, this is a resistant asexual spores that develop inside the bacterial cells. So usually, these spores are considered to be highly resistant and dormant with no metabolic activity or inactive activity. So usually spores functions for the resistant, I mean, of exposure of certain bacteria to higher or lower temperature. Okay, in order for the bacteria to survive even without food or even without nutrients. So sporulation is the process of forming spores. So Bacillus and Clostridium are considered to be endospores or spore-forming bacteria, and it is a means of survival whether moisture or nutrient supply is considered to be low. Okay, so we have here the sporulation process, wherein it is the process of endospore formation. So we have here the bacterial cells and chromosomes. So we have cytoplasm, cell wall, plasma membrane, and we have bacterial chromosomes or the DNA. Spore septum begins to isolate newly replicated DNA in small portions of cytoplasm. Usually, parang nagpo-form yan ng parang cleavage or invaginations. So, next, the plasma membrane starts to surround the DNA and membrane isolated in step 1. So, there is what we call separations of the cell membrane together with the DNA. Okay, and plasma membrane separates and encloses the DNA or surrounds the DNA. Next, spore septum surrounds the isolated portions, forming 4 spores. So this is considered to be two membrane protein. Two membrane structure. It is considered to be a isolation already of the forming of the spores. So peptidoglycan layer forms between the membrane. So this is what we call a synthesis of a protein, because peptidoglycan is composed of a glucose at the same time the peptides. And next, there is what we call a formation of spore coat, and formation of the spore coat followed by the release or the exit or efflux known as the endospores to spread from the cells. To free from the cell. Freed from cells, or kumbaga nag-exit, or other term na tawag yan na efflux. Kung pasulod naman, influx. Okay, so try to remember this usually happens in the endosperming bacteria known as Bacillus anthracis. Okay, so as a recap, so we have prokaryotic cells and eukaryotic cells. Summary, so mostly present prokaryotic cells are bacteria. There is no organelles present. There is new nucleus, unicellular, microscopic. So usually ang tawag doon nucleoid. Okay, and they have also DNA. Next, cytoplasm surrounded by membrane, and DNA is almost the same as eukaryotic cells. So they have organelles, nuclei. Plant cells, protocells, animal cells, spongial cells are considered to be eukaryotic cells. Okay. Next, for the reproductions of organisms and their cells. So bacteria commonly reproduce themselves very rapidly by the following methods. We have asexual, and we have genetic recombination or sexual. Mapaka na asexual, there is no sexual reproduction process. So there is no involved of the egg cell or a gametes, or genetic recombinations, and we have sexual. Okay, so these are the two reproduction process or bacteria of the cells. So asexual, it is a commonest method of multiplication process in the bacteria. Usually they follow binary fission, formation of buds, spores, cells, microcysts, fruiting bodies. Okay, so binary fission usually is the process in which genetic material of the cell is copied, then the parent cell divides. So this is very common in bacteria, yung tawag na binary fissions. Okay, so what this is the example of, or the diagram of the binary fission. So cell replicates to its DNA. So this is considered to a replication process or the copying of the DNA from the parent. Next, the cytoplasm membrane elongates and separating DNA and molecules. So cross walls forms of membrane invaginates. So this is formation of a cleavage furrow already. Okay, or invaginations. Then cross wall forms completely and formation and lead to the formation of two daughter cells. So this is simply the binary fission process. Okay. So another one is the formation of buds or budding process. Buds begins to form a parent cell. Usually this is what we call a bud that is formed. So nucleus copies and divides. Okay, that is a replication process. The bud receives a copy, but now becomes separate. But buds now are separated or become separate from the daughter, separate daughter cells. So budding produces chains of cells. So usually this is considered to be fastest also. So from one bud to another bud and to the it will form many buds. So this commonly multiplication process in bacteria.